Going live. You're live. All right. What's up, everybody? How's it going? We'll wait a minute or two, let people show up, and then we'll start talking about stem cells. Yeah, stem cells. Hey, Celia, what's up? Hey, Victoria. All right. See you, Pete.
I'll catch you guys tomorrow.
All right, catch you tomorrow. We got me and Jill here. We're ready to drop some knowledge on you guys and girls. How am I doing? Busy, exhausted. By the way, it's Dr. Zayner. I just want to, implore that upon you. I kid. You can call me Mr. Zayner. All right. So, if you saw in the Google Classroom, we're going to be, going over a paper today. The paper will help everybody get an idea of what it's like to read a paper. Understand a paper, and also we'll learn a bit about stem cells, which is fun, I think, because stem cells is something everybody hears about all the time. So I posted a link to the paper, so check it out. I'm kidding with you, Mr. Metric. Don't worry. You don't have to call me doctor. I find it funny when people call me Mr. Do I look like a Mr. Zayner to you? I mean, come on. What's going on with this? Yeah. So, what I'm going to do is... Can you guys see my face anymore? I hope not. Can I fit my face and the paper in the screen at the same time? My head might be too big. What we'll do is I'll cover myself and we'll talk about the paper. All right. Got the paper pulled up? You ready? The mic clips sometimes? Okay, let me try plugging in. All right, let me know if how the audio is now. Let me know if it's cutting in or out. Change it. All right, microphone now. Is it cutting in and out? Hello, hello. 1, 2, 3. Y'all know me, still the same old G, but I've been low key. All right, great. Nope, yes, no. Can you tell? You don't hear anything now? Ugh. Good Lord. Mike is showing up. I don't know why you're not hearing anything. Let's see. All right. What about now? Can you hear me? I hear you very, very quietly. You can turn up the volume on your computers, you know. This audio is too soft. Come on, people. Turn up that volume. Pump it up. Pump up the jam. All right, we're good now? Great. All right. Now we're going to talk about the paper. So pull up the paper. We're going to talk about the induction of pluripotent stem cells from mouse embryonic and adult fibroblast cultures by defined factors. All right. So generally, when you hear about science that is written about by mainstream media, all the mainstream media is doing is reading the summary, or sometimes it's called the abstract. Now, the summary or abstract tells you very little of what actually happened in the paper, but it makes it easy to sensationalize scientific research. So remember that. Just read the abstract. No, I'm just joking. As a scientist, as you get further and further along in your science, the things you care about more are just the 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 methods, because usually what you're trying to do is you're trying to learn from the scientific paper. You're trying to learn how to do a specific method and learn more about the way to do the science. So the methods are sometimes usually hidden all the way at the end, and they describe all the different techniques that were used, how they did the different techniques. It's also a good way just to learn science. Um, if you don't know anything about science and the techniques that are used, you can learn that in the methods, right? What's a Western blot? What's RT-PCR? What's a DNA microarray? What's bisulfite genomic sequencing? You go and you look up these things and you learn a bunch of different techniques and how to do science. It's great. It's perfect. But if we're trying to read a scientific paper all the way through from the first time just to learn about what was actually found, then we're going to read about the whole thing. You can read the whole paper all the way through. It's tough, it's boring, and it's usually better to print off the paper if you can. Honestly, I'll be honest with you. Majority of papers that I really want to read, I'll just print off. Um, it's way easier to read. I don't know why it is. There's just something about it. You could take notes, you can highlight stuff, you can just, there's something of having it all there in front of you where you can look at the figures and flip back and forth, where it's just really hard to do digitally. But yeah. All right. So what this paper did, I think it was the first paper where they turned normal cells, fibroblast cells, into stem cells. Now, what does that mean? What's a stem cell and what's going on here? So stem cell is basically this generic term that is used to describe cells that can turn into multiple different types of cells. So they call that differentiation, right? So when a cell can turn into another cell type or turns into another cell type, they call it differentiation. The cell differentiates into another cell type. Now, there's multiple different types of stem cells, right? So the the most powerful type of stem cell is called a totipotent stem cell. So you can just, you know, the word toti, um, I don't know if you know French, it just means all and everything, right? Like tout le monde, all the world. Toti means all. So totipotent stem cells are the most powerful type of stem cells, right? And they're generally only found in early embryos, right? Because, like, totally potent, it kind of does, Mr. Metric. It does sound like totally potent. So, like, totipotent stem cells, they can turn into a lot of things, but those are hard to get, and you're not really gonna use them or get access to them that often. All right. So the next type, most powerful type of stem cell is pluripotent stem cell. That's what they're going to talk about this paper. And don't ask me how they came up with these definitions or whatever. Pluri is like a plural, plurality. I can't say it. Plurality of different types. And then there is multipotent, which is the weakest type of stem cell, which can only turn into a few multiple different types. I don't know. I didn't come up with the terms, right? So when human beings use stem cells, right? So there's this big thing right now where people inject themselves with stem cells. They want stem cells to heal them, and they want stem cells to cure all their diseases. Well, the truth of the matter is, that's probably not going to happen. Now, stem cells, they can turn into other cell types, but all stem cells are not totipotent, right? Most stem cells aren't even pluripotent. Most of the stem cells that are used for injection are multipotent, or I don't even know what you call it if it's less than multipotent, but they're not that potent. And when you have cells that aren't that potent that you're trying to use, chances are they're not going to be able to help. They're not going to be able to turn into the cell type that you need that would help heal or treat a disease, right? But if you could, what you'd like to use is the most powerful that we can generally have access to, something that's pluripotent, because totipotent is, like I said, it's embryo type stuff. You're not going to get access to that. All right. So in this paper, they're trying to turn normal cells into stem cells. Why is that cool, right? Well, stem cells have to be harvested from someplace, right? They have to be harvested from an embryo or discarded cord blood from uh, eh, a baby or something. You have to get stem cells from somewhere. But if theoretically you could take, say, cells from your skin or your muscle and turn those into pluripotent stem cells, that would be amazing. So scientists started researching how to turn things into stem cells. Does bone marrow work? Does bone marrow work for what? What are you referring to, H.B. Wilder, in that question? Are you saying does bone marrow work as a stem cell in general? So generally what you're doing when you're injecting stem cells into yourself is you're injecting cells and you're hoping that those cells can turn into the cells near whatever you're trying to heal, right? And somehow heal that region. How does that happen? Mostly is fake and magic, but it can work and it can help, right? But you can imagine that, like, if you have, you know, a if you have a genetic disease, if you have a genetic disease and you inject yourself with stem cells, so if you have a genetic disease and you inject yourself with stem cells, nothing's gonna happen. This is not the way it works, right? That's not the way stem cells work. So what stem cells generally can kind of do is when stem cells get near other cells that are similar in nature, sometimes the stem cells can take on the nature of those cells. How would that help heal ligament tendon damage? I don't know. I don't know how it would help heal that type of damage. Don't ask me. But people think that it works. So, and maybe it does work sometimes, but it's not very scientific, and there's not much data to back it up, right? So when somebody presents something to you like this, stem cells can heal diseases. The first thing you should ask is, show me the money. No, show me the evidence that this actually does work. I don't want to hear that your friend kind of felt better. I don't want to hear that your boyfriend's sister's cousin got an injection and felt better, or you heard from some UFC fighter on Joe Rogan's podcast that they felt better. That's not evidence. That's not scientific. That's some anecdote that somebody says that could be placebo, could be anything, right? So ask for evidence. I'm not saying stem cells don't have any positive effect or can't have any positive effect. They definitely can. There are certain treatments that are FDA approved for using stem cells, but they're diseases. They're not for, muscle, ligament, tendon healing. And so there's that, right? So stem cells, be skeptical of it, but also be open that there's a chance that stem cells can do stuff for sure, right? But be skeptical. All right. So back to this paper. All right. So they're trying to reprogram these cells to be stem cells. So what makes a stem cell a stem cell? Well, a lot of it has to do, remember when we talked about the different DNA inside the cells and the DNA and the way the proteins are made, and how many proteins are made, and all these different things. These things relate to what the cell differentiates into and what it becomes. So if, say, we can find the things in the DNA, the proteins that are most responsible for stem cells being stem cells, then you can take a cell that's not a stem cell and maybe turn it back into a stem cell, right? That's the trick. That's the trick that they're trying to do. So they were trying to find what genes are responsible for a stem cell. And can we just add these genes to a normal cell and turn it into a stem cell? All right. So introduction, right? So then immediately you go into the results in a paper, right? So there's no like body. It's just like introduction, results. So for this experiment, what they did was it says that the results, if you're looking, we selected 24 gene candidates for factors that could induce pluripotency, right? So they found 24 gene candidates and said, all right, let's see what happens if we could take these 24 gene candidates and see if they induce stem cells, right? So how did they do that? Well, in science, you have to be kind of clever and tricky, right? Because how do you know if a stem cell is a stem cell, right? And what if you keep reading, what you can see is they say that there's this gene FBX15 that is is involved in stem cell maintenance in mice, right? So, but it's not required, right? The gene's not required, but it's involved in the maintenance of this pluripotency in cells. So if instead what we did was we put another gene in place of this XBF15 that we can have a readout of, we can see what's going on, then when our new engineered FBX15 gene is activated, we know that these cells are becoming stem cells, right? So you could use something like, nowadays they probably use GFP and a cell sorter or something like that. But in this case, they did something really simple. What they did was they put a gene in place of this FBX15 that protected the cells against a toxic compound. This toxic compound is G418. And so if you expose the cells to G418, cells that aren't stem cells are going to die because they don't have this new gene in replace of XBF15. They weren't turned into stem cells. But cells that are turned into stem cells will make this protein that provides them resistance to this toxin, and they'll grow. So basically all you're looking for is if cells live or not, right? It's pretty simple. So what they're going to do is they're going to take these 24 genes and they're going to see if we add these 24 genes in different combinations, do the cells live or die? If they live, we know that they might be stem cells because they're making this engineered FBX15. I keep messing that up. Sorry. It's complicated. Why can't they just call it like the Steve gene or something? The stem cell gene. It's a lot easier. Yeah, and that's what they did. So we go to figure 2, right? So what they're doing is they're looking for the the cells that are growing after a certain number of days, right? And you can see already that some of them don't do anything, right? The low colony number. Yeah, Mr. Metric, it is very much like using antibiotic selection. That method is very common. It's very common because it's easy to see. It's like a positive negative assay, right? Either things live and it worked, or things don't live and it didn't work, right? The live dead assay. It's really simple. So they're screening for these different factors, right? Seeing colony refers to like cellular colony. So that cells are dividing and growing, right? And so they added them, and you can immediately see some that have an effect and some that don't have an effect, right? But you can see that through different methods, what they wanted to see was um, they know that single genes aren't going to make the stem cells, so they need combination, right? So they need different combinations of these factors, right? So they tried all 24 together, and they found that works, right? So then what they could do is they could slowly reduce it down and see which ones work and which one doesn't. And they say eventually they found 10 different factors. And then from those 10, they tested those 10 more in depth, right? So basically what the process is a process of elimination, right? They have an idea, a general idea that these 24 different genes are responsible for something from previous studies, right? So that's why it's a good idea and important to, if you're thinking of doing a scientific experiment, you need to read up on the literature. Like what's going on, right? What have other people done? What experiments have they done in this area? You should spend a lot of time reading papers to try to figure out what other people have done because that makes your job so much easier. So much easier. That's how they got these 24 different things. And they narrowed it down and they kept narrowing it down. They kept narrowing it down until they found that 4 different factors alone could turn a somatic cell into a stem cell. 4 factors: OCT4, SOX2, CMYC, and KLF4, right? So we can see right here. So now what the rest of this paper is doing is verifying and proving, not proving, I don't like the word proving. It's verifying that the results that they have, the hypothesis that they have is backed up, right? So that's what most of a paper is. So most of a paper is like, hey, look, I have this idea of this thing, eh, and I'm going to try to provide a large amount of evidence in support of this idea, right? So if they were just to end the paper there after finding those 4 things, that would have been pretty good, but generally people would have said, well, we want you to do more in-depth testing. We want you to look at other things. So we want you to look at other stem cell markers using different techniques like RT-PCR and other things, right? So we want a more in-depth analysis of what's going on. We want you to do, well, this was back in the day, so they did microarrays instead of genetic sequencing. So they did microarrays to look at gene regulation. So microarrays were basically the primitive way to do large-scale sequencing. Basically, what it is it's DNA that's on a glass slide, and then what you do is you run other DNA over it, and if the DNA binds, it'll only bind to DNA sequence matches. So you could find like how much of different RNAs are in cells, how much genes are being expressed to give you an idea of what the global gene expression patterns are, right? Because certain cell types will have certain global gene expression patterns, right? So uh, uh, a stem cell will have different gene expression pattern than a cell that's already been differentiated into like a somatic cell, right? So back then, that's what they did. So they show the gene expression patterns of the different cells, right? And show they're different. Um, and then they actually try to turn these stem cells into stuff. So stem cells can form tissues called teratomas. So if you look at figure 5, it's the various tissues present in a teratoma. So a teratoma is kind of like a tumor, except it's a little stranger in that a teratoma differentiates into different tissue types. So sometimes people, maybe you watched weird Discovery Show channels and weird shit on there. I know I have. And they have somebody who's got like 100-pound tumor and it's got like teeth and hair growing out of it and crazy shit like that. Yeah, it's basically like a teratoma. So teratomas can differentiate into lots of different tissues and organs and crazy stuff. And that's the thing about stem cells is like we can't control what stem cells differentiate into necessarily, right? So if you inject stem cells into your leg to try to heal your ligament or tendon, like what makes you think they're just going to turn into like ligament or tendon cells? Magic? They're not always going to turn into that. Sometimes they'll turn into other stuff. There have been cases where people have injected stem cells. I think there was a clinic in Florida where they were injecting stem cells into people's eyes and they formed little teeth. The lady had like a tooth sticking out of her cornea or some shit. That's crazy, right? Because like stem cells don't all the time differentiate into what you want to differentiate them to differentiate into, right? There's a lot of things that control cellular differentiation, and it it's complicated. We don't exactly know how it works. If you had teeth growing in your legs, man, you could give some sweet roundhouse kicks, I bet, right? Yeah. All right. Yeah, so looking at what kind of tissues the cells can differentiate into, staining those tissues to, you know, isolate exactly what types they are. So they went pretty far in this experiment. Even so far, uh, where they created chimeric embryos. You're Mr. Metric, you're interested in stimulating tooth growth. Yeah, I don't know. I don't know, Mr. Metric. I don't know much about stimulating tooth growth, but it's definitely something that's possible. I just don't know if anybody knows how to control it, right? So that's something that you have to look into and figure out, right? Um, yeah, so most of this paper is taking this the stem cells that they created and then just verifying that these stem cells were healthy and functioning and working properly as stem cells. So they did karyotypes, they did Western blots to detect what type of protein. So a Western blot detects what type of protein is in the cell, if it's present, how much is present. So you can look up a Western blot and learn about the exact method. Ted, could you control the outcome of cell via culture? Yeah, you can. You can differentiate it in culture, but then it becomes more complicated. I guess you'd have to take your own cells, de-differentiate them into stem cells, then differentiate them into the cell you want them to be, and that's hard. It's not a really easy thing to do, and it depends on the tissue type. Some tissues are easier than others. And then the cells could maybe be used or helpful, but I don't even. So here's the question. It's like imagine if you had a cut on your hand, right? and you thought, oh, I know how I'm going to heal this cut. I'm going to chop off a piece of my skin and I'm going to put it on top of the cut. And that piece of skin is going to help that cut heal. Well, you know, it might a little bit. There might be some factors in that tissue that might help it heal slightly faster, but most likely you're probably going to have better luck just putting neosporin over that cut and it'll heal just as fast, right? So like the idea that just because a cell of a certain type is like you have an injury and you put more cells there is not necessarily going to heal it, right? Maybe. I mean, there are a lot of people who claim that stem cells have helped them for a lot of various different reasons. Um, so you can't exclude that it's a possibility how it works. But just scientifically, yeah, well, HB Wild, there are a couple of different things. So it's not just being able to differentiate stem cells. It's also um, being able to grow tissue in 3D. That's hard, right? So you have to imagine your organs are three-dimensional. So if you're just, uh, you know, trying to grow some tissue, trying to grow a liver, right? You can grow liver cells, no problem. But how do you grow a three-dimensional liver with the veins and with all the, you know, biliary ducts or whatever everywhere? Like, how do you make it so that it is the proper shape? Um, like you've seen stuff in the news lately where they grow little mini brains and stuff like that, right? And they can grow little mini brains. I think the stuff is the tour also had a tiny foot. What? That's creepy. That's really creepy. The tumor had a tiny foot. Oh, geez. Oh, good Lord. Mhm. Yeah, no, so like stem cells are a way to do this, and stem cells are a way to grow tissues in cell culture, and people are doing really new and cool things for it. Stem cells are great. I would just say, all the talk about stem cells out there, you just be skeptical. But I mean, that's just your job as a scientist in general, right? If you're trying to be a scientist in general, you should just be skeptical, no matter what it is. Who cares if it's stem cells? Um, you should be skeptical about it, you know, but be open. Be open. Yeah, no, Ted, it's really complicated, right? You can imagine that 3D structure forming, wow, is because it happens during development and there's so many different things going on, it's so complicated to try to recapitulate that outside an organism. The easiest way to do it is inside an organism. So a lot of people right now are turning to, can we create organs, say, inside a genetically engineered pig that's engineered to not have any antigens to the human immune system, right? If we can create a pig that has a similar, you know, blood type and organ compatibility to you, then you could just grow up the organs in pigs and transplant them into you or another animal, right? Xenotransplantation. That'll be pretty cool because outside of the cellular environment, it's like trying to grow up a steak, right? Cows are really good at making steaks, right? Because you don't have to like 3D print a cow. A cow just naturally grows into this three-dimensional object that can then be butchered and turned into steaks. Whereas if you're just starting with cells, like that's not very efficient. Imagine taking cells one by one by one and trying to turn them into a steak. Like that seems very difficult. Oh yeah, no. I mean, the thing is Kent, people do use scaffolding and all this stuff. It's just these are complicated issues and things, right? One thing I like the 3D print image. One thing to always remember as a scientist also, and not just be skeptical, but also if somebody hasn't done it, it's most likely not because they haven't thought about it, right? You have scientists who spent 20 years thinking about these different problems. So if somebody hasn't done it, it's most likely not because they haven't thought about it. It's because there's some other barrier in the way, right? So figure out what that barrier is. And then it opens up a lot of opportunities in scientific research. Oh, H.B. Wilder. Complicated questions. Complicated. So generally heart cells, when they're growing them in a petri plate and they beat, it's generally just normal heart cells that are cultured. But you could turn stem cells into heart cells also. They don't have to be subject to electrical charge either. They just beat on their own. Um, if an organ has various cells with other functions, how do they get a specific function? Yeah, no, that's one of the complicated parts, right? Is that some organs are composed of multiple different types of cells, different cell types, right? You have veins that bring blood into it. You have all these different parts and things that are composed of it. So it becomes extremely complicated, really fast if you're not just trying to replace one type of cell or one specific thing. Ted, that's a good question. That's a good question. Yeah, I don't know, but that sounds like an interesting thing to explore. If you are doing a stem cell injection or using stem cells, is there a way to expose them to specific things or specific factors or genes that limits the type of cells that they can grow into? Yeah, I think that's possible, and it's interesting. It is very interesting. It's clever. All right. So before we get too much into this, Jill's going to do a couple demos this week. So this week we're going to start getting into some more hands-on, make Petri plates. Um, and then from making Petri plates, what we're going to learn how to do is we're going to grow up some different cells, grow up bacteria and yeast and things like that. So from around your environment, after you make these Petri plates, we want you to culture different organisms, right? So you can put your fingers on the Petri plates. You can, wash your hands and then put your fingers on the Petri plate. You can, see what your toilet seat is like. You can see what your saliva is like, though I wouldn't recommend licking the Petri plates, even though they're not hazardous at all. Let's just you know, have a little bit of sanity. I think it'll just be salty because the LB media has salt. Not that I've tasted LB. Um, yeah, so culture different organisms from your environment because bacteria and yeast are everywhere, right? All over, everywhere. There's no surface that doesn't have it. They even survive in space. So now Jill is going to do a little demo and she's going to tell you a little bit about all this cool stuff. All right. So let's
Hey everyone, how's it going? So, uh, as Josiah said, you guys had three different things to read this week along with the paper.
oh crap.
Oh,
What's the wrong thing? Sorry about that. Hold on one second. That's what I meant to get rid of. Let me fix this really quick.
What do you do now? Yeah.
Configure video. I can't see what's going on. All right, we'll just keep it like this for now.
Okay.
Okay.
Okay, everyone. So we're gonna work on. There are 3 papers besides the scientific paper that you guys are reading on sterile technique, pouring agar plates, and culture. So we'll start with sterile technique. So of course we know put up our hair and put on our gloves. But if you guys already read through the culture part a little bit, the gloves we send you are pretty sterile, but they're not perfect. So we have some ethyl alcohol here, and isopropyl also works. And you just spray it on your hands and rub it round. And then just wait for it to dry so you don't kill any, doesn't drip into your solution and kill whatever you're trying to work with. And then you'll have nice, really sterile hands. So we sent you LB agar, which you poured in, sorry, not poured in. It has a concentration when you're making the LB agar plates of 35 grams to 1 liter, I believe. So here we tend to go with making 150 milliliters at one time, because that gives us about 7 plates, and it's what we do is standard. And so when you're making it, you go ahead, you pour the powder in. I'm not going to do that because I don't want to get Josiah's laptop all dusty and dirty and everything. Pour the water in, and then you microwave it. And you go the first time, maybe 2 times, you go 30 seconds, and the lid has to be super loose on top, okay? You just gently place it on because you don't want your bottle to explode in the microwave. And the one thing I think that people don't realize about this is you really have to take your time. And I do a little bit of stirring in between the microwave sections too, and I'll go down to 20 seconds and then even 10 seconds to try to not overboil it and have it boil over, but get it down so it's really clear. You want it to be quite clear. It might look a little miragey because it's pretty full of stuff, but it shouldn't be cloudy. The cloudy will give you the bad example plate, like there is on the agar culture, the agar pouring paper. So it should look pretty much like this, and then you want to cool it down really, really well so that you don't get condensation on the lid. If you pour it while it's pretty hot, it will condense on the lid. But I try to get it basically to the point of it'll almost start to gel up, and then I pour it in. Another thing is you want to pour them where you have just do 1 pour for the plate and then you're done. If you try to make it perfectly even, you will get lumpy plates, and that's not good. So it's, when you pour it, it's about that thick. This is a good thickness, so about that much. If it's too thin, it will dry up and maybe not cover the bottom of the plate, and that will be bad. If it's too thick, it's just a lot of agar to cool and kind of waste of agar. Plus, you don't want it to touch the top or get up on the sides. So once you pour your plates, a good technique is to stack them very carefully because it'll still be wet and moving at this point. If you stack them carefully on top of each other, then that will also help reduce condensation on the lids. Then after half an hour or a couple hours on the bench, you move them into the fridge, and we like to keep them there overnight so they are nice and solid by the time we want to use them. Because you do not want to try to put culture on a not completely gelled plate. It doesn't work very well. And then after that, we label them. If you don't have a lot in there, maybe it's not too much of a problem, but we label ours. So this is LB amp, so it's got LB and ampicillin. And then after it's gelled, if we don't use it right away, we store it upside down. And then that also reduces condensation on the lid. A lot of steps to do that because it's a pain. It doesn't look as pretty when it's done. And so that's most of it for the agar, pouring plates. And then on our culture little thing to have you guys read, there's basically a little mini experiment on growing bacteria, yeast, kind of what's on you with your fingers, which Josiah talked about earlier. And it goes with the unwashed hand, the washed hand, the gloved hand, and then the control. I do want to point out that on that paper, it's a split dish. So a lot of people last class, they were kind of quadding their petri dishes so that their control was a quarter of the dish, and then the non-washed hand and the washed hand were all quarters, and that could just have your hands too close to the control, something could fall off. If you really, really want to have a good control, you just leave it closed. I haven't opened this plate. It's not been opened since it was made, and just make it your control. That would be technically the cleanest thing to do. And then you can see if there's an actual problem with the control. Because we didn't give you guys split plates. They're kind of weird to work with. Yeah, that's pretty much it. I have to change this so I can see if there's any questions. Because right now I'm on the other thing. Okay. So let's see. 7 plates is how much Mr. Metric got. That's good. He labels it on the bottom. That works. Yeah, isopropyl is great. Ethanol is great. Paper. That's in reference to the paper. Hi, H.B. Wilder. Some. Mr. Metric has some icky looking cultures on one of the plates, and then his tap water has bacteria. I'm not quite sure what that is, but if you when you microwave your agar, it should pretty much kill anything in there. You get it basically to boiling point, and I don't actually know how long bacteria can last in the microwave, but it's not very long. Let's see, quartered one of the plates and disposited samples with a pipette. Okay, yeah, you can try it. If you want to quarter the plates, that's fine. So it's all just you guys testing it out and seeing what it's like. I overlooked it. What is the incubation? Oh, the incubation time. Actually, I'm not sure if it was on there. A week or so is good at room temperature. If you put it in the fridge, it will stall anything. So if you, for example, you're trying to make something cool looking with bacteria and then you have it to where you want it, you put it in the fridge and that'll kind of keep it in stasis for a good amount of time and it won't grow anymore. So a week on the countertop's good. If that's not working, 2, and then, yeah, then it should be good. Does general sanitizer help sterile? I think it leaves a residue. So It might sterilize everything, but then you could have this weird residue. Somo, common place to get pure yeast. I'm not sure what Somo means. You can get yeast from the grocery store, and we sell it at the ODIN. So getting pure yeast isn't too hard. Can you use standard convection versus microwave? I have done agar plates on the stove before, and it's kind of more of a pain, but you can do it. And oven, I don't know about people doing it in the oven. I don't see why not. You just basically have to dissolve the agar. So that should work. Okay. So that looks good. If you guys have any questions, we're always available on Google Classroom or by email. I'm Jill at the-odin.com. And then there's the Facebook group. We're still doing the Facebook group, right?
Kind of, yeah.
Okay, we're kind of doing the Facebook group, but if you guys message us on there, we'll see it. And we'll answer you. Are there any sources of contamination that could affect the results that are sometimes overlooked? Yeah, if you breathe too much on your blades, you'll contaminate it. The super sterile thing would be having a hood, but the vast majority of the time we're fine. As long as you're not like really bad, it should be okay. I live in El Salvador, so my room is about 30 degrees C. That should be it should work at that temperature, no problem. It will grow a bit quicker. So you don't need to cultivate it differently. It'll just grow a little bit quicker than other people's. Okay, so that looks like it's it. God, no seduction of place. We don't need that. So go ahead and try it out, and go ahead and post if you want on the classroom, and we can. We'd love to see your guys' pictures and how your experiments are going. So update us. And do you intend to sell autoclaves at the Odin store at some point? No, we don't use autoclave either. We use filters because they're heavy.
Yeah, generally you can get away without using autoclaves. And the majority of the experiments that you ever do in a home lab, you do not need an autoclave. You have to understand that like a microwave, when you heat it up, it's probably going to heat up your bottle to 150, maybe even 200 Celsius. And so that's going to be plenty to kill all organisms. An autoclave is overkill in the majority of cases. Like even when I worked at NASA and other places, I didn't use an autoclave. It's generally not necessary. It's just something that's kind of been ingrained in the minds of academic scientists, which is perpetuated to other people that you need autoclaves when you really don't. You don't need autoclaves. Just, be sterile and pay attention to things and you should be totally fine. You'll see when you make these LB agar plates, make a plate and leave one plate just as a control to see how sterile you were. Don't put anything on it and just leave it out. And it'll probably last pretty long before anything grows on it. You'd be surprised at not using an autoclave how sterile things are.
Okay, so our hour is up. Thank you all for joining our class, and we will see you next week. Bye.